हेलो गाइस, वेलकम बैक टू अनदर वीडियो। आज मैं बनाने जा रहा हूँ एक एडिबल सेल। जी हाँ, एडिबल सेल। ये मेरे स्कूल का प्रोजेक्ट था। दिस वाज अ स्कूल प्रोजेक्ट, सो आई थॉट दैट आई शुड मेक अ यूट्यूब वीडियो आल्सो showing that how I made it. So yeah, let's start. So first let me show you the jelly I made. Yeah, here, see, this is the jelly. So let's start first. First, I have to eat the Kinder Joy, so that I can take the chocolate part of it, the ball one, and I will then I dip it in Hershey's, and then I will set it for some time. That will be the nucleus of the cell. And to mention that I'm making an animal cell, so that's why the jelly is red. If I was making a plant cell, then it would be green in color, the jelly. So yeah, let's start. First, let's eat the Kinder Joy. I'm eating this like after 2 years. It's been the whole 2 years. I've eaten it. Yeah. So yeah, let me show you which part I need. You see this? I need this one, this part, just one. So yeah, yeah, yum. Yeah, this would work. Well, I've taken it out, one of them. So yeah, let's eat the rest. I really like chocolate, to be honest. Chocolate is my favorite. Well, I will not eat the second one also. Let's keep it for backup. So yeah, let's keep this as backup. अगर जमा नहीं तो एक और बैकअप भी होगा. There will be still another in backup. So yeah, I'll eat the chocolate part. Here are the two chocolate ones. I don't know what they are called. I guess they are hazelnuts. So yeah, these are the ones. Now I will now put them in a Hershey then I will dip in Hershey syrup here. So it will act as a nucleus in my plant. So yeah, just let me bring a hanky. Now let's bring a bowl and pour a bit of Hershey syrup in it. Then I will take this and dip in it, so that the Hershey syrup will act as a nuclear membrane which surrounds the nucleus.
A few minutes later.
So yeah, I have got a small bowl. Now let's pour Hershey syrup in it. So yeah, guys, I have the Hershey syrup ready. Now let's pour it. Let me bring it a bit up close. See, I'm going to pour it. Mmm, yummy. So yeah, I have taken the Hershey syrup, as you can see. Now let's dip these balls. Now let's dip them. Yep. Now let's put them in freezer for some time, or the fridge, so that it gets set. It gets set on it. Yeah. Just a second, a bit part left. So yeah, now let's set it. Yum. It's really gonna taste very tasty when I heat it. So yeah, I will tell you how it tastes in the end of the video.
A few minutes later.
So yeah, right now I'm freezing it in the freezer. So I'll see you after a few minutes.
A few moments later.
It's taking a bit longer than I expected. So yeah, till then let's cut the chupa chups, which will act as endoplasmic reticulum, the ER. But later in the video, I will also tell what are the functions. So yeah, actually I'm eating a chupa chups, this one, chupa chups built for first time, but I will not eat it. No, not this one. It's already been cut enough. Nope. Tastes better. Actually, not bad. Not bad. Let's see how this one is. Still better, but no. See, Yeah, it's in half. The one half side will go on one side, the other one will go on the other side. So yeah. So yeah, guys, it's taking a lot more time than I was expecting. So yeah, and I also bought another hanky. So yeah, let's wait. I'm thinking since it's taking a lot more time than expected. So let's see the let's see what toy I've got from the toy. So yeah, Superman. Well, if to be honest, I don't like DC much. I'm more of a Marvel guy. Yeah, let's see how to assemble it. Okay, Superman. Not expecting it quite well. Yeah. I'm not understanding it. So yeah, let's see. Let's see.
2,000 years later.
So yeah, guys, I think it's set. Yeah, it's completely set. So yeah, let's start. Now I will put this. Now I will put this in the middle of the jelly. So yeah, let's see if it can happen. Perfect, perfect, perfect. It's done perfectly. So yeah. Well, let's see the other one. Yum. Awesome. So yeah, it's perfect. Now let's put these. First, let me check if they are coming. Yeah, they are coming. So the first one, let's me try to put it in because I don't think it will come because it can be easy. Let me try. Let me break this part. Okay, so let me put it over in. But here, here. Yeah, the first one is in. Now, for the second one, I will put a little bit of network in. This will act as a ribosome, so this it will make this one rough endoplasmic reticulum. Okay, so yeah, and the other one was smooth endoplasmic reticulum. So yeah, let's open it. First, let's see the first one. Nice. Now, I will break it and I will sprinkle upon it. Okay. Yeah, I sprinkled upon it. Let's wait for 1 2 minutes so that it can stick. Perfect, 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 perfect. Thank God. It's perfect. Now let's put it in. Let's wait for 2 minutes. Yeah, jelly broke a bit. The jelly broke a little bit. Let me show you. Well, can you see it broke from here? So yeah, there's nothing we can do. But I have another gel, but first let me put everything. Then I can think of putting that above it so that it gets set. Then I'll put it in the fridge in for some time. Then it will get set and it will be ready. So yeah, let's continue. It's time for the gems, which will act as a vacuum. Vacuums are empty spaces. So yeah, in the animal cell they are very small, but in the plant cell, in the animal cell they are small and many in number. But in animal cell there is one big vacuole, very big. It occupies most of the cell. So yeah, let's take some gems. Proceed the first one. Oh, never mind. So as you can see, I've taken a few gems. Now let's put it in the jelly. Well, in the end, I will give a list of what things you need if you want to make an animal cell minimum. Not all things you have to cover, some of the organisms only. I will give a list of that. An animal cell, because plant cell is different than animal cell. So, let's take a few more. Let's put it here, here, no, not here, here, here. Let's become a bit watery. So, yeah, vacuoles done. So, yeah, let me remember what all I left. The cell membrane, the bowl, right, the nucleus, the smooth endoplasmic and endoplasmic reticulum, the vacuoles, then yeah, the ribosomes, of course, mentioned again. So let's tweak it a bit. Okay, now let's put it in places. Here, 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 here. So I guess it's done. So yeah, let's wait. Let's wait for some time. See what happens. Yeah. So yeah, actually now it's a bit watery. So I had made a jelly earlier, but it didn't turn out great. It became an ice cream. So I will bring that and I will place a small layer of it above. Then I will put it in the fridge so that it can set. Then it will be ready. Then I will tell all its functions, the food that which I use, what are they, uh, which cell organelle are they representing, and etc. etc. Then in the end, I will taste it and I will tell my honest review. Honest. Okay? A few minutes later.
A few minutes later.
So yeah, I have the old jelly here. It's actually completely frozen. Let me show you. See, it's completely frozen. So yeah, let's keep it in the room temperature for some time that it will freeze a little. Or I can just put it like that. Just like this. Yeah. No, I think I will have to wait a bit. Oh yeah guys, let me tell you that this might be our last video for I guess a few months. It will take a 1 2 months because my exams are due in September from 9 to 24. So yeah, I guess another video can come on Raksha Bandhan. That's on 22nd. But I'm not sure. So this might be the last video for some time. Then I'll upload another video which will be musical. So yeah, let's wait a few more seconds. So yeah guys, let me show you what I have done. So yeah, now it's like this. So I guess we can take this, put it on the cell I have made, this edible cell. Then I can freeze it for some time. I can put it in the fridge. So yeah, let's put it on it. So yeah, let's put it in the freezer for. Let's put it in the fridge for some time. Then we'll meet again.
The next day.
So yeah guys, it's in the fridge, the cell which I made. So I guess we have nothing to do except eat all this stuff. Yeah. Cheers. So yeah guys, it's time. I will tell you what the stuff I used and what they represented and tell you the function. So yeah, let's start. First, the bowl I used, in which I said the jelly, represented the plasma membrane. It is the outermost covering of a cell. It separates the content of the cell from its external environment. Yeah, it acts like a gate, so it can control that which molecule can enter and leave the cell. So yeah, let me give you an example. For example, our body, our human body, we inhale all the whole air, but it only allows oxygen to pass through the lungs, and it can only allow carbon dioxide to leave the lungs. It's like that. It's simple. It's made up of proteins and lipids, the plasma membrane. It is present in plant, animal, and bacterial cells. Yeah. So yeah, let's come to the nucleus. The nucleus is like the brain of a cell. Like our brain controls our body, the nucleus controls the cell. Yeah, it was discovered by Robert Brown in 1831. It is oval or round in structure and is usually present in the center of the cell. It's in the center of the cell. Yeah. So yeah, let's come to the cytoplasm. I use the jelly to represent the cytoplasm. It is the fluid part of the cell which is present between the plasma membrane and the nuclear membrane. Yeah, it contains various cell organelles. Yeah. Let's come to ribosomes. They are granular structures not bound by any membrane. They either occur freely in the cytoplasm or are bound to the rough endoplasmic reticulum. Yeah, let's come to the rough endoplasmic reticulum. I use the Chupa Chup bells to represent the endoplasmic reticulum, both smooth endoplasmic reticulum and rough endoplasmic reticulum. Yeah, rough endoplasmic reticulum. It has ribosomes attached to its surface, and it is used to synthesize proteins. While the smooth endoplasmic reticulum is in which the ribosomes are not present, giving it a smooth appearance. It is involved in lipid or fat synthesis. Yeah, let's come to the vacuoles. Yeah. Vacuoles, they are fluid-filled structures bound by a membrane present in the cytoplasm. They're actually empty holes. They have just fluids. In animal cells, they're small. They are small in size and large in numbers. But in plant cells, there is only one vacuole that is very big. So yeah, in plant cell, it covers almost 50% of the cell. Means it's very large. While in the animal cell, they're small, and lot in number. Yeah.
6 hours later.
So guys, it's set. So अब मैं अपना ऑनेस्ट रिव्यू बताऊंगा. ऐसा नहीं कि मैंने बनाया तो अच्छा ही होगा. परफेक्ट रिव्यू. और मैं सच बोलूँगा. और पहले मुझे कुछ दिखाने दो. देखो मेरी उंगलियाँ अर्ली होली या लेट होली कुछ भी कह दो हो गई. तो लेट्स स्टार्ट. मैं दिखाता हूँ एक बार कि कैसा दिख रहा है. जस्ट अ सेकेंड. सो गाइस ऐसा दिख रहा है थोड़ा थोड़ा. आई नो थोड़ा अजीब हो गया है बट क्या करूँ. चलो ट्राई करते हैं. नॉट बैड वेरी. अगर सच बोलूँ तो उतना भी बुरा नहीं है. अब और चीज़ें निकालते हैं. सो द स्मूथ एंडोप्लाज्मिक रेटिकुलम. इट्स नॉट बैड. एंड टू बी ऑनेस्ट मैं जेली पहली बार खा रहा हूँ. आई नेवर ईटन जेली इन माई लाइफ. सो नाउ कम्स द न्यूक्लियस. परफेक्ट है. सॉगी भी नहीं हुआ है. मैंने यूजुअली कभी कभी सॉगी रहता है. द वैक्यूल्स. टू बी ऑनेस्ट गाइस बहुत अच्छा बना है. मतलब मैं अपनी तारीफ नहीं कर रहा. ऑफ कोर्स हो सकता है कुछ लोगों को अच्छा ना लगे या ना लगे बट मुझे ठीक लग रहा है. आई फाइंड इट नॉर्मल. इट्स नॉट दैट बैड. या and if you will eat it after like a few hours it can get soggy हो जाता है. तो मतलब जब बनाओ ना थोड़ी देर सेट कर दो फिर आप खाओ वरना सॉगी हो जाएगा. थोड़ा मुझे लगता है. Awesome. Nutcut, ribosomes. ये थोड़ा सा वेयर्ड है. नटकट के साथ ये जेली थोड़ा सा अच्छा नहीं हो रहा है. एंड पानी हो गया जेली पानी हो गया पूरा. इट्स नाउ वाटरी. So guys, that is for this video. I hope you like the video. I hope you like the recipe. and I will meet you in the next video. I don't know when it can come on Raksha Bandhan, which is on 22nd, or it can come after my exam, which is after 24th of September. So yeah, goodbye.